Thanks, and hello everybody. Thanks for coming. It's a pretty good turnout. This is my first time giving a lecture to this group. I just moved from the University of Kentucky back in July, so I'm still pretty new here. Well, thank you very much. So, yeah, I know I support the Wildcats no matter what. Both universities have the same mascot, the Wildcat. Okay. And they're both equally good at football, apparently. So I'm going to talk today about molecular biology of brain tumors. This is what they asked me to talk about in the Brain Tumor Institute. The only problem is it's kind of a complicated subject. The field has changed a lot. We're talking about the genetic and molecular underpinnings of brain cancer, what different gene mutations cause different kinds of tumors. And it's actually a complicated subject, even to present to a audience of other physicians and scientists. This can be very difficult. And to an educated and informed but lay audience, it gets to be even more difficult. I want to make sure I'm presenting something that's actually useful and interesting, but you know, is not going to like bore you guys by getting too sophisticated here. And it's not because I'm any smarter than anybody else, it's just because I've made my career out of studying this stuff. So in cases like this, I always heed Albert Einstein's warning to make everything as simple as possible, but not simpler. That means, because he had the same problem trying to explain physics to lay audiences as well. Distill it down to a point where people can understand it, but don't oversimplify to the point where you start to lose the truth. And that's what I'm going to try to do here. So first of all, I figured it might be good just to start off with what a pathologist is. I mean, what is a pathologist? I am a pathologist, but what does that mean? What do I do? And then with that understanding, you'll have a better sense of just how revolutionary the molecular diagnostics have become in pathology over the last, say, 10, especially the last 5 years, and how it's changing the way we handle brain cancers. So first of all, there's these popular perceptions of pathologists in the popular culture, in the general nation, world as a whole, I guess. Pathologists are the one or two extremes. Either they're these creepy, cadaver-like guys who look worse than the bodies they're working on. Or, more recently, which I have to say as a pathologist, this is an improvement, we get these super educated, you know, good looking, smart, brilliant scientists who can tell what you've had for breakfast by your footprint. The truth obviously is somewhere in between, although we certainly like to think we're a little closer to this crowd than the other guy. Now, it's not, the fact that the popular culture doesn't get pathology quite right is all right. I mean, even to other doctors, we're kind of a black box. So the surgeon will take out tissue, and then it gets sent to some pathology lab, and within a few days they've got a diagnosis. But even most surgeons don't know exactly what's going on in between. It is really a black box. Now, to be a pathologist, first of all, like every other doctor, you have to complete a college and then pass the medical college admissions test. You have to do better than pass it. You have to do pretty well on it. And then you've got to complete medical school. That's 4 full years and pass the United States medical licensing exam, three separate steps, 1, 2, and 3, three separate exams. And then complete pathology residency, which is anywhere from 3 to 4 years, depending on the program. And then pass the general pathology board exam. Now, within the field of pathology, there's 2 major branches within pathology. There's surgical or anatomic pathology. That's the part that handles the stuff that comes out of patients or that does autopsies, that sort of thing. And that's the subspecialty in which I'm trained. And then there's clinical pathology, which is laboratory medicine. So when somebody has a blood draw, you have a blood draw and you get your calcium levels back, you get your red blood cell count back. That is being run by a clinical pathologist. And most of us then do, after all that, we still end up doing a subspecialty fellowship for another 1 or 2 years. In my case, it was neuropathology, but I mean, there's subspecialty fellowships available in areas you never even would think of, like pathology informatics, which is the study of big data in pathology, or forensic pathology is one, cytopathology, dermatopathology, pathology of the skin. I mean, anything you want, it's a big tent. The basic tool, this is a classic microscope from the 1880s. They really had style back then. It wasn't quite as powerful as what I've got, for example, sitting in my office right now. This is the same principle, but the microscope has gotten a tad more expensive and a bit more powerful. So this is actually something called a confocal microscope. This is sitting in my office. It's on a motorized stage. These are several lasers on the side that run lasers into the microscope so I can see things at really high magnification. And it's so sensitive, this is a special table that's actually sitting on a cushion of air. So if somebody's walking by my office or drives by us on the street outside, just this street outside here, the vibrations don't ruin my microscopy. It doesn't ruin the image taking I'm trying to do. So this costs more than a lot of people's homes. Just to give you some sense, I mean, science is expensive. But this is an everyday microscope, which is more classic. You got your eyepieces here, the slide goes down here, and there's another set of eyepieces in the back where somebody can sit opposite me and we can all be looking at the same thing. And yes, by the way, that is actually Luke Skywalker in a stormtrooper from the original movie, and not the prequels or whatever they call that rubbish. The good stuff. And in full disclosure, as long as we're going down this road, this is Dr. McCoy on the wall right next to my expensive confocal microscope. And right above my desk, I've got the bobbleheads, and I don't even know what this thing is, but I just saw it in the toy store when we were shopping for my kids, and I saw this. I'm like, I gotta get it then. You know, what? Gotta get it, yeah. I have no idea. But, ugly doll? Well, yeah, it is ugly. I mean, oh, it's called Ugly Dolls. Oh, okay. And to warn everybody about what they're about to see if they go inside my office, I even have something outside my door here. I got the Star Trek thing going on. And this special symbol is the medical symbol for Star Trek. I mean, I'm probably the only one in this room who cares. But anyway, I'm a pathologist, so they overlook that. This is the workflow of pathology, of general surgical pathology. This is the operating room. Most people here, unfortunately, are kind of familiar with that. And the tumors, the tissues that the surgeon takes come out of here, and then they send them to what's called the pathology gross room or surgical pathology suite. And the tissues are placed on the table like here, and then there's somebody standing there who will be working at it, studying it, take pictures of it, slice it up real nice and fine, describe it to log it into the medical records, and then places each little piece of tissue into these cassettes. There are cassettes about yay big, maybe about 1.5 inches in length, inch in width. And then you close the lid, put them in formalin fixative. That fixes and preserves the tissues. And then this goes to a nearby histology lab. Looks like any other lab in the hospital, where the tissues are put into this machine that actually embeds the tissues in paraffin. And it looks like this. This is the tissue. This is the piece of tissue sitting in this paraffin block. It's wax. It's just like, it's a petroleum product. It's like kind of a derivative of Vaseline, really. It's like candle wax. And then this paraffin block is put on a microtome, something called a microtome, which actually, when you mount the block on this little piece right here, and then you have this handheld crank that you turn. And every time you turn it, this moves up and down against a really super sharp blade sitting right here. And it cuts at a thickness of about 4 microns. That's 4 millionths of a meter thick. So it's very thin, very, very thin, that allows light to pass through, because if you can't have light passing through, then light microscopy doesn't work. Those little super thin pieces of tissue are mounted by hand. These are very special trained histotechnicians. They're like artists. And they mount the slide, they mount the tissues on these glass slides that are then put onto the microscope, stained, however you want to stain it, and then you can see whatever it is you're looking for. This essentially, even though the equipment looks fancy and it's all modern and it's nice and neat and clean, the process, the essential process, has not changed in over 100 years. Which actually, if you think about it, is pretty freaking amazing that guys in the Civil War period figured this out. They figured out a workflow that was unchanged for over 120 years. kudos to these guys. I mean, it's really phenomenal. But their reign is just about to end. And here's why. Because now we've got molecular testing, we've got a level of genetic testing that we can do to help us identify and diagnose tissues and diseases that light microscopy just can't handle. And here's an example of that. These are real cases that I encountered during my clinical practice. Here's an unfortunate 52-year-old guy who developed seizures out of the blue. It's never a good sign. And he went to a hospital elsewhere, and he had this MRI. He couldn't have an MRI, only a CT scan, because he had something in his back, something called the dorsal column stimulator. And he was treated with anti-seizure medications for 4 full days, and he was actually getting better. And so okay, we'll just maybe discharge him and hopefully figure out what's going on with him later out of the hospital. Well, they're just about to discharge him, and he crashed. So they did another CT scan, and this time he's got this. Now, before he didn't have anything on the CT scan, it was just a weird little blush. Now he's got this huge lesion that developed in 4 days. So, well, this is massive brain bleeding. If we don't operate on this guy right away, he's going to be dead in about 10 minutes. So they got in there and they took out a blood clot, and this is what we received as the referral center. This represents the entire specimen. This is 1 inch here, just to give you some sense of scale. Most of the red stuff you see here is just blood. That's all it is, it's blood. The diagnostic part ended up being about 1 millimeter in diameter. That's all we had to work with, not a lot. And under the microscope, traditional light microscopy, this is what we saw. These sheets of cells, these are all little cells here. This is all blood. Here, all this stuff is blood. The red stuff is blood, but these little guys are cells. And we really weren't sure what it was. We weren't even sure it was a tumor. We're looking at this, we're like, well, what do we do with it? We can't call it a tumor. We can't call it, we don't know what it is. So I said, all right, let me try. I run the molecular lab. Let's try a few genetic tests in case this is what I think it might be. If it's a glioma, we got some tests that can prove it. One of my favorite tests of all time is fluorescence in situ hybridization. This is, long story short, you can actually synthesize little stretches of DNA artificially and tag them with these neat little chemicals that glow. They fluoresce under the light, right, the correct kind of microscope, like the microscope I got in my office with all the lasers. And these pieces of these DNA probes, these fluorescently labeled DNA probes, will bind to specific spots within the nucleus of whatever it is you're trying to study. And you can design the probes to bind to whatever you want. So in this case, we used probes that were designed to bind to a specific gene called EGFR, also known as epidermal growth factor receptor. This is a gene that is a very powerful gene. It's turned on in a lot of cancers, including glioblastomas. And it's matched up with another little, it's matched up with something called chromosome 7. The gene is sitting on chromosome 7, and there's another special fish probe that binds to chromosome 7. The bottom line is a normal cell will have 2 copies of chromosome 7, because you're supposed to have 2 copies of every chromosome, every gene in your body. So you get 2 copies of chromosome 7, that equals 2 green signals. You have 2 copies of EGFR sitting right next to chromosome 7. I mean, it's on chromosome 7. So you should only have 2 copies of EGFR, 2 orange-red signals. So in this case, though, when we got the tissue, this is what we found. This is a 1000x magnification. These are the cells. First of all, a lot of these cells have 3 copies of chromosome 7, 3 signals. No, there's no non-cancer that gives you 3 copies of a chromosome, all right? This only happens in cancer. And in particular, what really drove the point home was all this, while it's projecting, you can see all these orange-red signals. They almost look like a cloud. Instead of 2 orange-red dots, now we've got so many you can't even count them. Well, this is actually what's known as EGFR amplification. This is, instead of having 2 copies of EGFR, you can have 50, 100, 200, 300 copies of EGFR. There's only one, in the setting of a brain tumor, there's only one kind of tumor that does that. That's glioblastoma. It actually happens that about 50% of glioblastomas have this, and it sticks out like a sore thumb on fish, the fluorescence in situ hybridization, which is what makes it such a handy test. So sure enough, based on just that molecular test alone, not on light microscopy, but just based on that molecular test alone, we were able to diagnose it as a grade 4 glioblastoma. And unfortunately, the patient did die within 4 months, in keeping with the aggressive nature of this disease. Now, that's not a very uplifting story, but at least in this case, we were able to give him an answer. Him and his family were able to give them an answer. You know, up until then, they actually thought he had a brain bleed caused by high blood pressure, because his blood pressure was a little high. And that's what they thought was going on here. We were able to show, no, actually, it's not the case at all. What he had going on was a brain cancer. So now his family knows, and they have some closure. And he was even able to set his affairs in order and spend the last 4 months of his life in some comfort and rest in hospice, instead of bouncing back and forth with doctors constantly trying to figure out how to manage what they thought were strokes. So the diagnosis in this case ended up being a form of therapy. Now, granted, we want to do better someday, but at least it's a start. And it was a prominent enough case. This was a case I handled back when I was at the University of Kentucky. It actually made the cover of their UK Healthcare On-Call magazine because it was really considered a high-profile case. But they're not all bad news. I mean, molecular diagnostics is sometimes actually can give you really good news. This was a great story of a 14-year-old kid who had a tumor in his thalamus. Now, that's a deep-seated part of the brain. It's like right in the exact dead center of your brain. Very hard to get to surgically. And the tumor that they were able to get out under the microscope, it was diagnosed as, well, it looks like a glioma, but we really can't tell you anything more than that, other than it doesn't look malignant. It looks like a lower-grade glioma. But it wasn't very satisfying to anybody, because low-grade gliomas, especially in that location, can still kill a kid just as well as a high-grade glioma can. Because if it keeps growing back and everything, and then they can't control it, there's plenty of kids that will die of these low-grade gliomas. And this is what it looked like under the microscope. Is even to me, looking at it now, I can't tell you what kind of tumor this is by light microscopy. It just looks, all I can say is it doesn't look malignant. Well, a different FISH test, the other one was EGFR FISH. This is a different gene called BRAF. And these are the tumor cells, and you can see a slightly different pattern here. Now we've got 2 green signals and 2 red signals, and then there's this third little red signal in a lot of these tumor cells. And I'll give you a high magnification look here. So you got these 2 green signals, 2 red signals, and then this third little guy sitting next to one of the larger red signals. That actually means something. I'll explain what it means in just a minute, but at this point we're saying, okay, this is positive for BRAF rearrangement. That's the term. So how'd the kid do? Well, this is a little bit of a trick I'm playing, because this case was actually in 1986, about 20 years before BRAF testing was routine, at least 20 years, and he's now 40, he's older than me, and he's still alive. That tumor never grew back. That was the end of it. He was cured. So what is it? What was that thing? Well, as it turns out, BRAF, the gene called BRAF, is actually in the same, this is really wild, it's in the same class as the EGFR gene. But when the BRAF gene is turned on, yes, it causes a tumor, but those tumors usually end up burning out after a certain length of time. They just stop growing, and they just sit there, and then eventually they wither away. It's kind of weird that way, but that's how it works. That's why the cancers are complicated. And we can detect, and it's also not turned on by amplification. So in other words, you don't get many copies of BRAF. For whatever reason, that doesn't happen. Instead, you get this gene rearrangement where normally you get 2 copies of BRAF, and then for whatever reason, you get this screwy rearrangement where you end up with a third fusion gene. So part of BRAF fuses with this other gene called KIAA1549. There will be a quiz, so you better remember that. The thing is, we don't even know, nobody even knows what this gene does yet. But for whatever reason, it pairs up with BRAF, and this little nubbin of BRAF, it's not even the whole copy of BRAF, it's just a little nubbin of the BRAF gene, ends up as part of this fusion gene, which encodes a fusion protein. You can actually see it by FISH with a 3-probe cocktail that covers the entire part of the BRAF, and so you end up with these big 2 large BRAF signals corresponding to these 2 guys, and then this little nubbin right here, which corresponds just to that little red part. The long and short of it is that when you see that, it's actually, by and large, it's a pretty good indication that this kid's going to do well. Because the studies have shown that well over 90% of kids with this gene rearrangement end up cured. They don't need radiation, they don't need chemotherapy, they don't need anything. So like I said, it's not all bad news. And it actually helps us, because then when we know somebody's got that kind of a gene rearrangement, They're a little less likely to treat the kid with radiation chemotherapy. You know, they're not going to be as aggressive. They want to see if this thing will burn out on its own, which especially in a kid is what you want to do. So the long and short is that the times, you know, to quote the Bob Dylan song, as everybody does eventually, the times they are changing. So you've got, it's no longer acceptable just to look at these different tumors under the microscope and say, okay, that one's a glioblastoma, grade 4. That's a medulloblastoma, World Health Organization, grade 4. This is a ependymoma, grade 2, whatever, that's lovely. That's not good enough anymore. Now we have to generate, in addition to that, we have to generate all of these incredibly complicated genetic reports to say, okay, yeah, you've got a glioblastoma, but actually this one is driven, instead of EGFR amplification, this one's driven by a mutation in a gene called H3.3. Again, don't worry about it, but it does actually have a different prognosis than one that's driven by EGFR amplification, and hopefully someday it'll have a different therapy too. Same goes for medulloblastomas. We now know there's 4 different groups of medulloblastomas. They can all look exactly the same under the microscope, but there's actually 4 different genetic subtypes of medulloblastoma, and they have different behaviors. They have different prognoses. And then finally, these ependymomas, which also occur in kids. I mean, there's 2 different types, with and without this fusion in the gene called REL-A. Again, the details aren't important, just to recognize that the same tumor, 2 different tumors can look exactly the same under the microscope, the same light microscopy we've been using for 120 years. They can look the same under the microscope, but they're driven by different genetics. And if they're driven by different genetics, the prognosis can differ radically. Some of these kids end up, or adults end up surviving for years. Maybe they're cured. Some don't make it even a year. Now, the 2 examples that I gave you were strictly focusing on this one gene, one test approach. That's the traditional approach. I showed you 1 test for EGFR, 1 test for BRAF. But now the technology's advanced so much that it is now economically feasible to test many different genes all at one time. And one of the ways we do it is through something called next generation sequencing. This is where you take this, a stretch of DNA, and you just sequence it and pick up all the mutations and gene rearrangements and everything else that's screwy in the genome. It used to be prohibitively expensive to do that. I mean, you just couldn't do it on a routine basis. But for example, when I was finishing up my PhD in the year 2000, they just had sequenced the entire human genome. It cost $1 million to sequence 1 human genome. Now you can do it for $1,000 and get better data than you did 15 years ago. It's incredible. So I developed a panel with my colleagues from the University of Pittsburgh, where I trained. And for the first time ever, this panel is now actually available for Northwestern patients, brain tumor patients. It's a 30-gene panel that hits all the major high-yield mutations in all the different major classes of brain tumors, gliomas and meningiomas and medulloblastomas, all these different grades, adult and pediatric glioblastomas, and really everything that we handle now surgically could potentially benefit from this gene panel. Again, 30 different genes, all of which can be diagnostically and prognostically relevant, can dictate therapy, and we can do it for the cost of about $1,500, which, if you know anything about the cost of medical care, that's barely a rounding error in the total cost of taking care of one of these brain tumor patients. It's very, very cheap compared to the amount of data you yield from it. And that's actually not even the most cutting edge stuff. The most cutting edge stuff hasn't even come out yet. I mean, there's early papers, but it's not available on a routine clinical basis yet. It's something called methylation fingerprinting. And I'll just throw it out there because this is going to be the next big revolution in molecular genetics, probably in the next 5 years, I'm guessing. So you got these stretches of DNA, and everybody knows about that double helix with the DNA. You've seen that before, but it's not just sitting there in a vacuum. Actually, in the cell, this DNA is being modified heavily by certain pieces of certain chemicals that are being attached to various parts of the DNA. And one of those chemicals is methyl groups. In other words, a little carbon with 3 hydrogen atoms bound to it. Well, that little methyl group, that CH3 group, will bind to different spots along the DNA and radically change the DNA's behavior in the cell. What's really fascinating is that this methylation, every cell in your body has DNA that's methylated or not methylated. And the pattern of methylation throughout your entire cell's genome is unique to that particular cell lineage. In other words, it acts as a fingerprint. So if you have a clump of cells and you don't know where they came from, you can actually do DNA methylation fingerprinting, and at a 10,000-foot view, it will generate a pattern of methylation and non-methylation throughout the entire genome that tells you what tissue it came from, what tissue those cells came from. This is brand new stuff. And it's really handy. It's great for research purposes. It's fascinating for research purposes, but even on just a pragmatic level, even if we don't know exactly why the methylation patterns are different, we can still use them to help classify difficult tumors. Metastatic tumors is actually one of the most common kinds of tumors we get in neuropathology. In fact, they outnumber primary tumors. In other words, tumors that grew in the brain, that arose in the brain, are outnumbered by metastatic tumors from the lung, from the breast, from the pancreas, wherever, by about 10 to 1. So you get 10 times as many metastatic tumors as you do primary brain tumors. But many times we can't tell where the metastatic tumor comes from. Sometimes the patient has a metastatic tumor and you're like, and they do CT scans of the entire body, they can't tell where that tumor came from. Methylation fingerprinting can help tell you where that tumor came from. It can also help classify primary tumors that are otherwise really tough to tell on the light microscope. Here's another case. This is a 13-year-old boy who has seizures, and you see this huge tumor that was growing in the front of his brain. This is the front of the brain, this is the back of the brain. And long and short of it, under the microscope, I can't tell you how many hours I spent staring at this crazy tumor under the microscope. All I could tell the clinicians at the time that this was a high-grade tumor. And it looked malignant. In other words, I was worried that this was really going to behave aggressively. And that's as far as I could go with it. I couldn't tell what kind of tumor it was. And nobody else got me. I even sent it to my colleagues at Johns Hopkins. They had no idea what it was. I showed it to a few others. They couldn't tell. We did all kinds of special, expensive stains and studies, and they didn't help. Without going into the gory details, these are the major possibilities. This is the problem with it sounds like every time we issue a diagnosis that it's absolutely, this is absolutely what it is. End of story. Written in stone. But the reality is a lot of the tumors that we have to diagnose didn't necessarily read the textbook. They don't play by the rules. They can look like a little bit like one tumor, but a little bit like another tumor. So what do you call it? You got to call it something. And these are the different kinds of tumors that this thing actually kind of looked like. But they all have radically different prognoses. They're all treated differently with different chemotherapy and everything. So the clinician's like, well, what do we do with this? This was a tough one because at the time, this was like, a year and a half ago, maybe 2 years ago now, I guess. We didn't know what else to do, so I advised them and said, look, if the family's interested, we can send this tumor down to Baylor. They have a huge genomics laboratory down there. And they'll sequence the entire genome of this tumor. It'll cost several $1,000s because they have, for clinical reasons, it's a little more expensive. But I said, there's nothing else I can do with this. And even then, I warned him, I said, there's only a less than 50% chance that it's going to actually yield anything useful for us. Because actually a lot of times the mutations, you look at the mutation results and you're like, well, okay, it's got a dozen mutations in this tumor, but it still doesn't tell me what this tumor is. So we did it, the family did it, and they spent quite a bit of money, and sure enough, within about 3 or 4 months, we got the results back because it's a slow turnaround process. And disappointed to say that we weren't able to get the diagnosis based on the sequencing. But at least they knew that there was that risk. And I swear to God, it couldn't have been more than a week after I got those disappointing results from Baylor that a friend of mine from New York University, molecular neuropathology is a small world, and so we talk to each other a lot. And he emailed a whole bunch of me. He says, look, I got this new test, this methylation fingerprinting. If anybody's got a case that they are having a hard time with, send it to me and I'll do it for free. I'm like, I think I can help you there, sir. So I sent him the material, I sent him the pathology material, and within a week, they get this result. And this doesn't look like anything. And the bottom line is, it definitively said that this tumor is something called a primitive neuroectodermal tumor, Wnt subtype. Don't worry about what that means. All I'm saying, this is so cutting edge. This entity does not yet even exist in the World Health Organization textbook on brain tumors. That's how out there we're talking. However, based on this, and the results weren't even close. I mean, the score, don't worry about what score means, but there was, that's what it was, because there was nothing else that was even close in the final algorithm that the computer generated. It said, it's got to be this tumor. So I said, well, all righty then. That might explain why I didn't know how to diagnose it under the microscope, because it's a really experimental subtype. But based on the genetic pattern, this tumor should be less aggressive than it looked like under the microscope. That's what these tumors are. This Wnt subtype of tumors actually can fool you. It can look more aggressive and it can look nastier than it really is. Well, sure enough, followed this kid up over a year later. I think it was about a year and a half now. He's still got a little bit of tumor sitting in the resection bed of where it was operated on, but that's it. It hasn't grown back so far. Now, maybe it will someday, but it's a lot better than originally. I thought he wasn't going to survive a year based on what that thing looked like under the microscope. But the genetics said, the methylation fingerprinting said, no, actually, this guy's he's got a chance to live a little while. And sure enough, he's doing good so far. So the bottom line here is that our understanding of brain tumor genetics has absolutely exploded in the last 15 years, especially in the last 5 years. It's getting to the point where it's hard to keep up. And it is improving our diagnostic and prognostic accuracy, as you've seen. But we still have to be very careful on how we interpret the results of each of the tests. I mean, it's not a panacea, and there are still cases, even after genetic testing, that we're not entirely sure what to do with the tumor. And therapies that are designed to target specific genetic aberrations, the true personalized medicine that everybody's looking for, that holy grail, is coming, but it's going to take a little bit longer. And we kind of all knew that was going to be the case. It's going to take a little bit longer for that sort of thing to come along, but at least we're on the right track. So this idea of pathology being a black box, well, hopefully to you guys, it's a black box no longer. The only thing is now it's, whether that's a comfort to you or not,